especially when you know the sequence of that piece of DNA. And the method basically employs a few important ingredients or reagents. It requires tiny little pieces of synthetic DNA that you can synthesize in a machine that match the sequence you want to amplify, part of the sequence you want to amplify. And then it uses an enzyme that works at very high temperatures, usually around 65 or 70 degrees. And it needs nucleotides. And it of course needs the template that you want to copy and make many copies of. And the process basically works as follows. If you have a piece of DNA, and there is a segment of this DNA that you want to make many copies of, the first thing you do is you're going to denature this DNA by heating the DNA to a high temperature, 95 degrees, 96 degrees. The DNA will become single-stranded. Once the DNA becomes single-stranded, you're going to add these little synthetic fragments of DNA called primers. These DNA fragments are perfect matches to the region or to the ends of the region you want to amplify. So you make these little oligonucleotides that are usually 20 nucleotides long, 10 nucleotides, 15, 20, 25, 30 nucleotides long. These little segments, they are perfect matches with this DNA that you're trying to make a copy of. One will, of course, be 5' to 3' in one direction, the other oligonucleotide will be 5' to 3' in the other direction. So you denature the DNA you want to amplify. You add these little oligonucleotides or primers into the mixture and you reduce the temperature to 55 degrees. So you lower the temperature. At this temperature, these little fragments of DNA will hybridize based on sequence. Once these primers hybridize, you then initiate by adding a DNA polymerase. Now this is a special DNA polymerase that functions at a high temperature, 70 degrees centigrade. And in the presence of deoxyribonucleotides, these will get this enzyme, which is a DNA polymerase, not an RNA polymerase, will basically replicate the DNA using one of the strands of the template. And it will make a copy of that, incorporating these nucleotides during the process of synthesizing the second strand. So you basically initiate synthesis, and the two strands that were denatured and hybridized with these primers are basically going to tell the DNA polymerase, here is the point from which you should start synthesizing DNA. And so the DNA polymerase will basically initiate synthesis and make a second strand of DNA to make this DNA double-stranded. And so you've now gone, for this one segment of DNA, you've gone from one copy over here, double-stranded DNA, to two copies of double-stranded. Then you repeat the cycle again. You denature at 95 degrees. So you have 4 strands of DNA. You add primers, they will anneal or hybridize to those segments. It's the same primers as before. They will hybridize. And then the polymerase will synthesize a new strand. And now, after 2 cycles, you've gone from 1 copy to 2 copies to 4 copies. And then you repeat the cycle again. And you will end up with 8 copies. And then again, and you get 16 copies and so on. And you usually go through around 30 cycles and make the enzyme go through the same set of sequences 30 times. And you end up with over 1 million copies of the region of that DNA that you want. So it's a very quick, efficient method to amplify a particular fragment. Repeat this cycle over. So here's what it actually looks like in practice. Imagine you want to amplify this segment that's labeled in red. And you want to make many copies of this segment. So what you need to do is you need to make primers. And the primers, where you design the primers is up to you. You could have the primers in the region that's black. And it will make a little segment that's black as well as you could design the primers right in the red segment. It's up to you. The choice is yours. And depending on what function you want to use this for, you design the primers range you want. Because these primers are made basically by chemical synthesis in a machine. You can design them the way you want. So let's imagine we design a primer that has the sequence AAAGATC. This little primer will hybridize to this strand over here, the black segment, 5' to 3', 3', 5. And the second primer that you design will read 5' to 3', DAG, ATCGA. That's DAG, ATCGA. So we have designed two primers that are flanking this red segment. We denature this double-stranded DNA. There you go. And this primer will hybridize to the top strand, right there. While this primer will hybridize to the bottom strand of the DNA. Now, just like RNA synthesis goes from 5' to 3', right? Messenger RNA is always made 5' to 3'. When you replicate DNA, DNA is also replicated 5' to 3'. So the synthesis, the direction of synthesis between the RNA polymerase and the DNA polymerases are the same. The difference, of course, is in RNA polymerases, you have a specific promoter, and the polymerase is recruited to the promoter via various proteins. In DNA polymerases, it's this little primer that will recruit the DNA polymerase enzyme, and this enzyme will then synthesize the second strand. So the primer is basically initiating the start of replication. And so you end up with replication taking place. It goes 5' to 3', and it will replicate this strand of DNA, making a copy using complementary bases. Similarly, you make other strand. And so that's how you will make a copy. And then, of course, you repeat the cycle over and over and over again, and you will get many copies of this segment in red. It will have those extra 10 nucleotides on either side, making the primer sequences. But in effect, you will have amplified history. Well, any questions on this? If I were to give you the floor.
When does it stop?
It stops when it falls off the end of the DNA, or the length of time you let it go for. So you usually, rule of thumb, not perfect, but rule of thumb is, let's imagine you want to make a copy of a fragment of 1,000 phosphate of 1 kb. You amplify it for 1 minute. It's approximately the speed of the amplifier is kind of considered to be 1 kb. And so it only amplifies for about 1,000 base pairs before the reaction stops, because you can denature the DNA by raising it again to 95 degrees. That's one way. If you don't do that, you let it run for 10 minutes, it will run for as long as it can and ultimately falls off. The enzyme is not as processed as your normal enzyme. This is an enzyme we isolate from specific bacteria. Actually, it's not even bacteria, it's archaea. From hot reds in Yellowstone. Those are the original archaea that were isolated. And it's from there that we isolated this enzyme. So the enzyme is not very processed, so it runs for about 4 to 5 kb, maybe 10 kb, and then it literally falls off the DNA on its own, and then it stops. So the point is, if I were to give you a piece of DNA like this and ask you to design a specific primer, would you be able to design it? You have to learn to remember that when you design these primers, the primers have to go 5' to 3'. And they have to be on opposite strands, so they have to match the right one. Okay, so how do we detect the PCR reaction? Well, we detect the PCR reaction by running them on a gel. Just like we were doing before with restriction fragments. The PCR, the region that we've amplified, because we've made many copies of it, we can put it on a gel, we can run the gel, we have a marker, of course, and we ask what is the size of the fragment that we have amplified. And if the fragment is small, you see a band running at this pad. If it's larger, it will run here. If it's very large, it will run size. So, size of the PCR product will depend, of course, on the location of the primers. You can have primers over here, you get a small fragment. If you have primers here and here, you get a larger fragment, and so on. So the position of the primers determines the fragment that you're going to have. Right? Make sense? And then you basically detect the size of these fragments by running them. The gel is simply a way of detecting the fragments, the size of the fragments. The gel.
Yeah.
Obviously, because the fragments run based on size. And so the smallest fragments run the fastest, the largest fragment runs the slowest. So you can clone these fragments of PCR. So you can PCR amplify them. Once you've made many copies of this DNA, you can clone them by putting them into a plasmid platform. Now you have a copy of that PCR fragment on a plasmid, and now with E. coli can make many copies. So there are two ways in which you can do this. You can just keep amplifying it in the lab in a test tube. Every time you need more of this fragment, you just go and do a PCR reaction. That's one way. Alternatively, you take that PCR fragment and throw it into a plasmid, and now let the bacteria keep making many copies. So there are two ways in which you can amplify a PCR. By cloning, by PCR, or by PCR combination. It depends on what you want to do. So when you have it on a plasmid, it's almost like a permanent pad. You can freeze it in the -80 freezer for 20 years and nothing happens. And it's in a bacteria, you just have to pull it out and make many copies. When you have to do it by this, you have to have the oligonucleotides. And then you make copies just enough for the work you want to do. So you don't have them as a perceptual copy. So it varies. It depends on what you really want to do. If it's something that you're only going to use once, there's no point in cloning. So then you just do a PCR. Okay, so now we're going to turn to a method that allows you to analyze a specific single piece of DNA without cloning. And that's known as BLOF. This was a method that was developed in the late 70s by a scientist, I believe, I'm going to get to guess. It's in England. He's from England. I'm going to say Manchester or Birmingham, one of those middle England cities, but I may be wrong. His name was Ed Southern, and the method is called Southern BLOF. It's named after the person who discovered it. So we were able to make a restriction map of a piece of DNA after we cloned it. We got a plasmid, we got a piece of DNA, we got many copies of this piece. So once we had many copies of this piece of DNA, we could map that piece of DNA using various restriction methods. And we got a very nice detailed map of that fragment of DNA using restriction mapping. And we've seen how to do that. So the question is, can you restriction map a piece of DNA without cloning it? That's the question. And the answer is yes and no. You need to have cloned this fragment of DNA at least once. So somebody somewhere has to have cloned that piece of DNA. Let's imagine one person in this class is being very altruistic and clones the piece of DNA that you are interested in. But you want to analyze the restriction map of that piece of DNA in your own genome, not that other person's genome. That's the question. Can you determine the restriction map for an unknown piece of DNA using a known piece? And that's what Southern does. So, it's a rapid method for studying a specific DNA fragment from a mixture of fragments, so long as a part of that fragment has been cloned by someone. In the assay, let's take a simple example first, and then we'll go into a little bit more complicated situation. Let's imagine there is a plasmid, so it's a molecule of DNA. And let's imagine you cut it with a restriction enzyme. and you get two bands. Right. You have two fragments now, because you've got it at two split places in the gene. One of these fragments contains a gene, the other fragment doesn't contain a gene. But you don't know which of these two fragments contains the gene and which of them does not, fragment does not contain the gene. So, there's a 50-50 chance one of them is right. That's not good enough for us. We want to know for sure which of these two fragments is the fragment that contains the gene. That's the question. So, you have an unknown mixture of fragments, and in this mixture, there is one fragment that you want to identify, that you are interested in. Now, to do this experiment, you need a probe. That's a fragment, small piece of DNA for the gene that you are interested in. It doesn't have to be the entire gene. It can be a small fraction of the gene. 100 base pairs, 500 base pairs, some things, a small region. That's all you need. Once you have that, you'll be able to identify which of these fragments is specific. Okay. So what you do is you take this mixture of DNA and you run it on a gel, actual gel. So here is your marker with different size fragments of known size. 1 kb, 2 kb, 3 kb, 4 kb, 5 kb, 6 kb. Here is this plasmid that's uncut. Run over here. And then when you cut it, you get two bands. Right? But you don't know which of these two bands contains your gene of interest, whatever gene you're trying to study. So, how do you determine which band corresponds to the gene? That's the question. Okay, so we use a probe to identify that fragment. So, you take the gel that you've run, and then you transfer it to a membrane, a filter paper. The reason is the gel is three-dimensional, correct? It's like a slab of gel. So it's soft, it's fragile, tends to break very easily. And the DNA is buried in a three-dimensional motion, basically a slab, and the DNA is inside. So it's not easily accessible. Yes, you can diffuse things into the gel, but it's not easy, and the gel will break very easily. It's very kind of gooey. You know what Jello feels like, right? It's exactly that sort of feeling. And so it's not easy to handle when you're trying to do this experiment. So the thing you do is you transfer the DNA from the gel onto a piece of paper. And it's done as a perfect match. So you're basically, literally transferring the DNA from the gel onto the paper by blocking. Oh, this is very easy. Here's your gel, right? And the DNA is basically as a band in the gel. Wow, this is perfect. This works. Okay, so here's the gel. Now what you do is you take a special membrane. This membrane basically does not allow the DNA to go through. It's impermeable to DNA. Okay? But liquids will go through. Buffer water will flow right through. But the DNA will hit this membrane and stop. So the DNA is right now in this gel. What you do is you put the membrane on top of the DNA. You put a wad of blotting paper. So this gel has liquid in it, correct? Jello has liquid in it. And you basically put this wad of blotting paper on top. Here's your wad of blotting paper. And you go home for the night. You come back in the morning, all the liquid has been sucked out of the gel into the blotting paper. And as the liquids move from the gel up into the blotting paper, the DNA moves with the liquid. But it hits the membrane and stops. Right? So now you have your DNA stuck on the membrane. And now it's accessible, and it's on a membrane that's not going to rip or break very easily. So you have the DNA right on the surface of the membrane. It's stuck. It won't fall off. That's the most important thing. And it's sitting there, ready for you to analyze. So you transfer the DNA from the gel to a membrane, which is a microcellulose. It's exactly the same thing as what we used to blow up proteins, microcellulose. Okay. TME, etc. So you have to be a little careful. You don't want to take this piece of membrane and put it in a flame. It goes poof in your face. That happens occasionally. And you get into trouble. So you don't want to do that. But anyway, that's a different story. Transfer the DNA to the membrane. So here it is. So remember, the DNA is on a particular. This is the germ. Here is the marker. Here is the man. We're going to put a membrane over here like this. Right. So, don't look. Okay. Yeah, never mind. Anyways, so here's the gel. The DNA is in the gel. We're going to transfer the paper like this, and you're going to turn it over. Right? So you're going to get a mirror image of the original gel. Correct? Because you flip it over like that, because that's what the surface of the paper is, where the DNA will be stuck on. Right? Here so far, wasn't too confusing. Okay. So here is the membrane with the DNA transferred onto it. Then you add the probe. So the DNA is on the paper and it's DNA. So you basically nature the DNA. That's very easy to do. You take the membrane with the DNA on it and you soak it in sodium hydroxide. Sodium hydroxide will break the double-stranded bonds, the hydrogen bonds between the bases, and the DNA becomes single-stranded. So it's attached to the membrane, but it becomes DNA. At a very micro level, the DNA becomes single-stranded. Now you take this filter, and to it you add radiolabeled probe. Question.
Why does it really sit together so long?
It's the charge. The way the paper is and the way the DNA is, that it doesn't allow it to flow over. If you absolutely can, there are ways in which you can make the DNA flow through, but it's very inefficient to the point where, for what we are doing, it doesn't. Okay, so now to this paper, we're going to add a probe. This is a small piece of DNA that matches the gene that you're interested in. It's only maybe a fragment of the DNA that you're interested in, but it has to match it. And you have artificially made it radioactive. So you take the membrane with this piece of DNA, like this. You take a tray, you put the membrane in, you put some buffer with this radioactive probe onto it. All right? Now we're going to go on to some water. Here we go. Let's imagine now this is our gel, and we have DNA that is separated on the basis of size. So we have 3 pieces of DNA separated on the basis of size. If I could hold them like this, you could see this is my gel, and there are pieces like this, right? So there is a piece that's green, a piece that's yellow, and a piece that's blue. This is on the membrane at specific spots like this. Correct? And then I'm going to add some probes. I'm going to add a probe that has a sequence that matches the yellow. So I'm going to add it onto the membrane. I have the membrane, I add the probe onto it. Again, You let it walk away for a night, overnight. It just sits around, the probe's looking for its own perfect sequence. Based on sequence, it will find the match and it will hybridize to it. So it will form double-stranded DNA. Remember, the DNA has been denatured. You're adding this probe. This probe is also single-stranded DNA. And you're going to ask it to find its perfect match based on complementary sequences. When it finds that band, right there. So out of the 3 bands, that yellow probe that you added will find only one of those 3 bands. It's the second one. Correct. Now, you take that membrane with the radioactive probe and expose it to an X-ray foam. Because that yellow band is radioactive, the probe was radioactive right here, that band will get labeled. That band will get labeled, and it will show up as a specific band on the X-ray foam. So when I transfer this, so here is the piece of DNA. Here is the sequence of the DNA. It goes GATC, 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 TTTT, GATC, GATC. And my little radioactive probe is labeled AAAAA, but it's radioactive. It's emitting beta rays. Okay? So it's emitting gamma rays or beta rays. That's really what you use, or even alpha rays sometimes. We label this piece of DNA. This DNA will hybridize. It will only hybridize to this fragment because that's where the sequence is. It won't hybridize to this fragment because this sequence of TTTT is missing on this fragment. This fragment has some other sequence and it doesn't have a match. So this probe will only hybridize to this on the DNA, on the membrane, right there. So this probe will get hybridized and it will become radioactive on the membrane. Now when you expose that to a filter, to an X-ray foam, you will see that only one of the 2 bands will light up. The second band will not light. So you transferred both bands, only one of them will be radioactive and will give you a signal on the X-ray foam. And so that's how you will be able to say that this fragment, out of these 2 fragments, is the one that has a perfect match for the probe I was interested in. Is this clear? Yes? Sort of? Yes, go ahead.
I'm confused at how it picks where to die, or how it picks where to attach.
It attaches based on sequence complementarity. So what you do is you hybridize, so you take your membrane and a buffer, correct? And we leave it at basically 60 degrees overnight with the probe. So at 60 degrees, the probe is binding and coming off everywhere. It's trying to match itself by base complementarity. And it will, once it forms a perfect match at 60 degrees, it stays stable. It doesn't come off. And that's the key. It's to get that right temperature where the probe will bind only its perfect sequence, but not an imperfect sequence. If it hits an imperfect sequence, it falls off and goes looking again for the right sequence. When it hits the right sequence, it stays bound and never comes off. And then that's it. Then it stays labeled. And so you label this one fragment out of a mixture of fragments.
So if you make this probe right,
Correct. That's right. You make the probe. So that's the whole key. The whole key to this experiment is getting the probe. And that's why I said one person in this class has to be altruistic and has to clone that piece of DNA. And that takes a bit of work. Doing a Southern blot is not as much work as making the probe.
Just to clarify, going into this, you know the sequence of DNA already?
You know the sequence of that little segment of DNA. Not the whole DNA, but just a fragment of it. That's what you need to do. Okay, so the probes are obtained by a number of different ways. You can get RNA and make that into a probe. So, for example, let's imagine you're interested in the globin gene. Right? The globin gene, where is the globin gene made? Red blood cells. right? Why are red blood cells red? Because it has the globin, hemoglobin in it, right? So the red blood cells are humongously enriched for the globin transfer. If you make RNA from red blood cells, the vast majority of the RNA is globin messenger RNA. So you can take that messenger RNA and you can radiolabel it. Now you have a probe